So, students, we will talk about community today. So, what is community and what is community ecology? The study of different kinds of population in an area is called community ecology. And when a group of several species like plants, animals, or any microorganisms live together with mutual adjustment and beneficial interaction in a natural area, it is called community. As you know, that this is biotic in nature, means community is only living in nature. Organisms share the same habitat, growing in uniform environment like forest, grassland, a desert, or a pond. This is type of natural communities. So, as I have told you, a community must include only living entities of the area. But if abiotic part is also considered, we would be dealing with an ecosystem. So I will tell you these terms in next slide. A community has its own structure, composition, and developmental history. So I would like to introduce few things like in a hierarchy, individual or any organism is a basic unit. So when a group of same organisms living in a common habitat interacting with each other is called as population. Population means जनसंख्या या आबादी. When different types of population live and share the same physical space, it is called as community. Community is samudaya. This is always biotic in nature, means living. In a community, if we consider the abiotic, means non-living things, then the whole structure is called as ecosystem, और जिसे हम पारिस्थितिकी तंत्र भी कहते हैं. And finally, all the ecosystems of the earth together make the giant ecosystem, and it is called as biosphere. Biosphere means Jeev Mandal. The concept of community is not so recent, and it may be traced back to the time of Theophrastus. He first time recognized the existence of plant communities. And later on, in 1838, Griesbach told that plant is an important unit of vegetation. And Edward Forbes in 1844 studied the mollusks in sea at different areas at different depth. Then Carl Moebius in 1880 coined the term bicoinosis. And Schroeter and Krejci in 1902 introduced the term synecology, which is the study of population. So population study is synecology, and when we study about an individual, its life cycle and everything, then it is called as horticology. So the size of population is referred to as population growth, and it may be affected by birth, immigration, and other factors. And factors that limit the population growth are called as limiting factors. So what are the characteristics of a community? This is species diversity, growth forms, dominance, succession, and trophic studies, that is the energy. So each community has its own composition, structure, and developmental history. And in each community, there are different diverse groups. So in composition, we can see that communities vary in size. This may be large or small or very small size. So in large community, we may include forest, desert, extended over areas of several thousands of square. And in small communities, like meadows, river, ponds, rocky plates. And in very small size, that are called as microhabitat too. So microbes on a leaf or log or in litter soil is called as very small size communities. So what is the basic structure of community? So besides composition and dominance, community exhibit a structure of recognizable pattern, and this is arranged in a spatial manner, thus may be divided horizontally into sub-communities. So this is a unit of homogeneous life forms and ecological relation. And if we consider about the zonation, then the horizontal division constitute the zonation in the community, and this is the example of horizontal zonation. So you can see that at the fit of 13,000, the tundra biome is found, and below that, it is started from 2,000 feet. You can see the desert biome is here. So this is the long range, and this each community, the whole community, is divided into many horizontal zones. You can see the conifer zone, the coniferous pine zone, and the tundra zone. So if we see in a shallow ponds, zonation is very little, but in deep ponds जो बहुत गहरे होते हैं, लेक्स और जो तालाब होते हैं, उनमें जो हॉरिजॉन्टल प्लेन्स होते हैं वो विजिबल होते हैं. एंड द अपर वन इज कॉल्ड एज लिटरल जोन, मिडिल वन इज लिम्नेटिक जोन, and the lower most is called as profundal zone. So this is the horizontal zone. But if we talk about the vertical zones, we can see in forest. So another aspect of structure is stratification, which involves the vertical rather than horizontal changes within community. Sometimes stratification is very complex, where a community possesses a number of vertical layers. So in grassland communities, subterranean floor and herbaceous stratum, only two zones are found. So in subterranean floor, basal portion of vegetation such as rhizomes of grass covered by litter and debris of animals and plants, it is found there. But in herbaceous stratum, means some herbs and grasses are found. And in a forest community, which is very large, the strata is complicated. And in general, 5 vertical subdivisions are there. So the lowermost is subterranean subdivision, and then forest floor, that is the earth surface, then herbaceous vegetation, means herbs, grasses and everything, then shrubs, shrubs means undershrubs, shrubs and large shrubs, and then trees. In trees, small trees, trees and large trees, all are included here. So based upon the light and relative humidity, this may be 8 subdivisions, because if we consider about the underground part stratification, roots and rhizomes are also present. So third characteristics of community is the development, and this is called as ecological succession. And in this we will deal in the next lecture, that is ecological succession. And then you can study here some part, that process of seedling establishment and successful growth is called as excesses. The result in the formation of colonies in new areas is called as colonization. So these are different community theories: the time theory, the spatial heterogeneity theory. Third is climate stability theory, productivity theory, competition theory, predation theory, and stability time hypothesis. Now, what are the different methods to study the community? So first is the floristic method, and second is the physiognomic method. So these methods are not now in common use. The first method and are descriptive methods. Here, vegetation is described in terms of flora. So listing of various genera and species present in the community is the flora, where naming of plants is also done. And this gives a little idea of composition. But physiognomic methods, in this life forms are considered, and it includes the study of growth forms, general structure, its spread. So the most studied this method is by Ronquiers, and Ronquiers studied in 1934, and his study concluded such descriptions that I will include in the later slides. What are the different characters used in the community? To study the detailed aspect of any community, like species diversity, growth forms, structure, dominance, and its successional trends, a number of characters are used. For example, if we say that this particular species is dominant, means that that particular species or plant exert a major influence on that community, which finally determine the nature of the community. कि उस कम्युनिटी या उस समुदाय का क्या नेचर है, ये डिपेंड करता है कि वहां की डोमिनेंट स्पीशीज कौन सी है. So these various characters used for such purposes are broadly classified into two major categories. First is analytical character and second is synthetic characters. So analytical character is expressed in terms of 5-point scale, and these characters are frequency, density, abundance, cover, basal area. And physiognomy, phenology, stratification, abundance, sociability and vitality. So these are all included in analytical characters because it is divided into two, quantitative and qualitative characters we will study later. And second is the synthetic character. Here three characters are used. First is presence and constance, second is fidelity, and third is dominance. So we will deal with analytical characters, and this is further classified into two categories. First is quantitative, which I told you. It is expressed in quantitative term, frequency, density, abundance, cover and basal area. And second is qualitative, which is expressed only in qualitative way, although finally again grouped in 5-point scale. So, quantitative characters. The first one is frequency. Frequency is the number of sampling units in which a particular species occurs. So, different species of community are recorded by various phytosociological methods, and the frequency of each species is calculated by the following formula. So, percentage frequency is the number of sampling units in which species occurred upon total number of sampling units studied into 100. So, this is the frequency of each species we can calculate by this. And when Ronke studied, he concluded the 5 frequency classes. So, these are different frequency classes A, B, C, D, E. So, on his extensive study, he concluded that A is greater than B, B is greater than C, C is equal to, greater or lower than D, and D is always lower than E. So, second is density. Density represents the numerical strength of the species in the community in any unit area. It gives an idea of degree of composition. It is calculated by the following formula. That is total number of individuals of the species in all sampling units. These sampling units may be a quadrat upon total number of sampling units studied. So, this is the numerical strength of the community. And third is the abundance. In abundance, the formula is by total number of individuals of the species in all the sampling units upon the number of sampling units in which the species occur. So, means it is density upon frequency. Values such as frequency, density and abundance do not provide the correct information on the distribution of a species in a particular area because various species differ in their growth forms. Therefore, such values are to be supplemented with other characters like cover and basal area. So, cover is used in general sense for the area occupied by a plant, may be herbaceous cover or basal area, used for the above ground parts like leaves, stem and inflorescence. And the basal area is the ground actually penetrated by the stem and is rarely observed when the leaves and stems are clipped at the ground surface. So, basal cover determines the dominance and is measured by calipers, line interception or point-centered quadrat method. So, this is grouped into 6. Group 1 tells that less than 5% if cover, and in group 2 5 to 25% cover is found, and in group 3 25 to 50%, group 4 50 to 75%, in group 5 75 to 95%, and in group C 95 to 100% cover. B is the qualitative characters, and this falls under the category of analytical characters. Here the first is physiognomy, general appearance of the vegetation as determined by the growth forms of the dominant species, like larger trees are dominant with some shrubs would tell as a forest. So this tells about the growth forms. And second is the phenology. It is the scientific or systematic study of seasonal change, means the periodic phenomena of organisms in relation to their climate. A study of date and time of events is phenology. So phenology is the calendar of events or the life history of the plant. You can see the events as a phytogram. The first one is germination, second is the vegetative growth, third is the flowering, fourth is the fruit formation, fifth seed maturation, and sixth is the death. So the third one is stratification. Stratification is the way in which plants of different species are arranged in different vertical layers in order to make full use of the available physical and physiological requirements like sunlight, etc. And it can be obtained by a bisect the vertical strata in a community. Stratification is the result of interdependencies of species like lianas and epiphytes that grow on other plants. So example of liana, you can see Tinospora cordifolia, and this is also called as Giloy or Guruch or Amrita. So you can see the figure of liana, that is Giloy, and this is the stem and whole part even. It is a medicinal plant and this grows in the dense forest, and to get the sunlight, it reaches on the top of the forest. Fourth is the abundance. Although related with density, it may not be expressed in quantitative terms. Organisms, particularly plants, are not found uniformly distributed in an area, found in smaller patches or groups, and they differ in number with each place. Divided in 5 groups, that is very rare means very less abundance, rare, then common, then frequent, then very much frequent. And the species which are very much frequent means almost dominant in that area. Fifth is the sociability. Sociability means proximity of the plants to one another. So species with some density, it may differ in sociability values. And sociability expresses the degree of association between species, how these are associated with each other. Some divide the sociability into 10 classes, like Brown-Blanquet in 1932. He divided into 5 groups. The S1, this is the first group, is where the plants found separately from each other, thus growing singly. And second group, a group of 4 to 6 plants at one place. And in S3 group, many smaller scattered groups at one place is found. And in S4, several bigger groups at one place is found. And in S5, a large group occupy the larger area. And sixth is the vitality and vigor. Vitality is the capacity of normal growth and reproduction that is important for successful survival of a species, depend upon the weight of species. In plants, stem height, root length, leaf number, and leaf area, weight of flowers, fruits, and seeds, these all determine the vitality. So on the above criteria, plants are grouped into different vitality groups by R. Mishra and J. S. Puri in 1954. R. Mishra, as you know, he is the father of modern ecology. And in these 3 groups, the first group is well-developed plants completing their life cycles regularly, and they produce the viable seeds. And second group, plants reproduce vegetatively. And third, ephemeral, short-lived. The plants are short-lived. That may be called as seasonal also. And Brown and Blanquet in 1932, he gave the 4 groups, and Daubenmeier in 1968, he gave the 5 groups. So these 5 groups are, the first group is the plants capable of germinating but dying soon without reproducing. In second group, vitality group, plants which determine life for some time but able to reproduce. And in third vitality group, plants reproducing vegetatively. In fourth vitality group, plants reproducing sexually but somewhat feebly. And in the fifth vitality group, plant reproducing sexually very well. It shows the response of the area is very good. And this is the major second group is synthetic characters. So these are determined after computing the data on the qualitative and quantitative characters of the community. For comparing the vegetation of different areas, community comparison needs the calculations of their synthetic characters. So these are determined in terms of first is the presence and constants. This expresses the extent of occurrence of the individuals of a particular species in the community. How uniformly a species occurs in a number of stands of the same type of community. The species on the base of its percentage frequency may belong to any of the following classes. The first is rarely present, means it is present in 1 to 20% of the sampling items. Sampling items may be a quadrate or any method. And second is the seldom present, means 21 to 40% of the sampling unit. And third is the often present, in this 41 to 60% covers. And fourth is mostly present, so these species cover 61 to 80%. And fifth group covers the constantly present area, means 81 to 100% of the sampling items. Again, this may be a dominant group. Second is the fidelity in synthetic characters. Fidelity is the degree with which a species is restricted in distribution to one kind of community. So here is restriction. These species are sometimes known as indicators also. The species have been grouped into 5 fidelity classes as follows. You can see the first fidelity group is plants appearing accidentally, means sometimes plants appear in fidelity. Second group, indifferent plants, and it may occur in any community. In fidelity third group, plants occur in many communities but predominant in one, and this is called a preferent. In fidelity fourth group, specially present in one community but may occasionally occur in other communities as well. So these plants are called as selectives. And in fidelity 5 group, these occur in only one particular community and not in others, and these plants are called as exclusive. And third of the synthetic character is dominance. Dominance is described under quantitative characters. Here the dominance is expressed in synthetic form also. So on the basis of density, frequency and dominance, cover values, there has been proposed the idea of importance value index, IVI. It is also called as IVI. So IVI of a species in the community gives the idea of its relative importance. For IVI, values of relative density, relative frequency and relative dominance are obtained as follows, and this formula is used. You can see in relative density, the density of the species upon total density of all species into 100, and to find the relative frequency, the frequency of the species upon total frequency of all the species into 100, and to find the relative dominance or cover, the dominance of the species upon total dominance of the all species into 100. So we can find all three characters, and if these values are added, IVI values of different species are arranged in decreasing order. And other synthetic characters are, which have some value in further defining the community, are interspecific association, association index, index of similarity, dominance index, species diversity and diversity index. So here I end the first part, and in the next lecture we will talk about other things like life forms and other many things in community part 2. Thank you so much.